to you by Lipid Maps.
Welcome to the Lipid Maps Podcast. I'm Matthew Conroy, and today we're joined by Professor Frank Löffler, who holds the Goodrich Chair of Excellence at the University of Tennessee. Frank, welcome. Thanks for having the time to talk to us.
Well, thank you very much for giving me the opportunity and having me here.
Pleasure. One of your research focuses is what we often call forever chemicals, the polyfluoroalkyl substance. Can you explain what they are and why you're interested in them?
Okay, so per and polyfluoroalkyl substances, PFASs. So these are compounds that have unique properties because the hydrogen atoms on the carbon chain are substituted by fluorine atoms. We use them in various applications, household applications, many industrial applications, military applications. They are everywhere. You know, when you go home, you have them in many different products. I give you a few examples. You know, when you take your Gore-Tex jacket and it repels water really well, and it also is stain resistant to oily compounds, it's PFAS. When you spill something on your couch and it doesn't penetrate the fabric, PFAS. When you clean your teeth with floss and it glides really well, PFAS. When you go skiing and the wax on your skis works really well, it has been PFAS. So the stuff is everywhere. It has become apparent that these compounds are recalcitrant. We find them in environmental systems. We find them in every person's blood. Every person on Earth has PFAS in their blood. People are concerned about this, and there's a strong push to remediate, get rid of these chemicals, but this has proven to be very complicated. And there's also a strong push to reduce production and application of these compounds. And I think it's certainly doable. You know, there are certain things that we can replace them easily. You can wax your skis with other products. You can replace them for many applications, but not for all. There are applications where PFASs are essential. So, for example, the computer industry wouldn't function without PFASs. You would have to give up your phone. And this I would probably call essential applications of PFASs. This is a struggle right now where, you know, regulators and industry has to come to a conclusion. The bottom line is that these chemicals have penetrated the environment, and we are concerned that they have negative impact on human and environmental health. I should say potential impact because toxicity to humans has only been demonstrated for a few of these chemicals. When we talk about PFASs, we have to realize it's a class of compounds. There's hundreds, thousands of different PFASs at any given contaminated site. And the theoretical number of PFASs in environmental systems, right, just theoretically from the production process, millions. Right, so this is not one compound. These are many different types of compounds that share this common structural element of perfluorinated carbon atoms. For that reason, now we are interested in, you know, what happens to them in environmental systems. And as a microbiologist, environmental microbiologist, the obvious question is, what can microbes do?
Yes. Yeah.
And this is how I got into this research theme. There's funding available to explore PFAS, fate and transport in environmental systems. And this is how my research started on these compounds maybe 6-7 years ago.
And you showed recently asking what microbes can do with these compounds. You had a paper in Nature Microbiology that showed exactly one of the things they can do with those compounds, which is put them in their phospholipids. So what inspired you to look there?
Yeah, so this is an interesting observation. There's a story behind it. So I'm not a lipid guy at all, right? So I have the basic understanding of lipids from when I was a student. You know, I had a professor who was very keen on lipids, so I knew a little bit, but then I forgot. So we had an organism, an isolate, a Pseudomonas species from garden soil that could grow on terminally fluorinated alkanes, C10, C8, you know, C12. And when this PFAS business became more interesting for researchers, everybody started working with PFASs. And I thought, you know, maybe it's more productive to work with model compounds. And so the idea was, let's try if this organism can degrade terminally fluorinated alkanes. So not PFASs, but terminally fluorinated alkanes as model compounds. And, the organism could grow on these compounds. It could release fluoride. Okay, interesting. There's a monooxygenase, an alkane monooxygenase that attacks the fluorinated carbon and releases the fluorine substituent as inorganic fluoride. It actually turns out that this monooxygenase has a preference for the fluorinated carbon over the methyl carbon. Monofluorodecane, so only one end fluorinated, it likes the fluorinated carbon more than the methyl group carbon to do the attack. Okay, so then the organism releases inorganic fluoride. And I had a student who was very careful with mass balance analysis. So Jamie Xi, he's now a professor at Nanjing University in China. And so he measured fluoride very carefully. And he always came back and says, I can't close the mass balance. I can't close the mass balance. Something is missing. There's fluorine missing. And since we could very nicely quantify the substrate and the inorganic fluoride, we were looking, where does this fluorine go? And I had an opportunity here to work with the mass spec facility on campus. We did some metabolomics studies. And so we started with a C10 alkane. And then we observed in the metabolome fatty acids that were terminally fluorinated that were longer than C10, suggesting to us there must be some biosynthesis going on. Biosynthesis, that includes fluorinated metabolites. And so then we had the hypothesis, if we have these long-chain fatty acids, maybe we can find them in the lipids. And we started to do lipidomics. And indeed, we found glycerophospholipid species with a single fluorine substitution in an acyl chain. So this is different from what was published in this Nature Microbiology paper. I'm telling you about this because we developed the lipidomics approach that then helped us to make this discovery that entire polyfluoroalkyl substances can be incorporated. So this is how we developed the lipidomics approach and were able to measure fluorine in membranes. And then the student published this work in Environmental Science and Technology. And then he did an experiment where he took the organism and he tried to grow it with different PFASs. And it didn't work, didn't grow with PFASs. But he could grow it on acetate and tested if different PFASs could be co-metabolized. And then he made this observation that when he added 8,3-fluorotelomere carboxylic acid, so 8 means the number of perfluorinated carbon atoms, 3 means the number of carbon atoms that are not fluorinated. So the total chain length was 11, 8,3-FTCA, and he saw some consumption and then asked the question, what happened? And he had this lipidomics workflow available and applied that. And to our surprise, he came back and said, I find glycerophospholipid species where one of the acyl tails or sometimes both acyl tails were replaced with 8,3-FTCA. And that was, okay, interesting, but there were lots of questions, to verify. I mean, it took years, to verify that this is really the case. We worked with a separate mass spec facility and let them also work on this to verify the existence of these molecules. And they all came back. It was very clear in the end that yes, this bacterium incorporates fluorotelomere carboxylic acids, basically recognizes them as fatty acids and directly incorporates them into the glycerophospholipid pool.
So this accounts for the missing mass that you were saying before.
So now these are two different processes I just described. The first observation with the terminally fluorinated alkanes, if you have a fluorination on both ends, the organism removes the fluorine on one end, does beta oxidation, generates fluoroacetyl-CoA, which goes into biosynthesis to synthesize longer chain fatty acids that the organism then incorporates into the glycerophospholipid pool. Okay, and that's where we made the observation about the mass balance issue. We couldn't close the mass balance. After that, the student did an experiment, right, to ask, can this organism co-metabolize PFASs? So now we talk about H3FTCA, not about fluorinated alkanes. And here the H3FTCA is not defluorinated. The organism takes it as it is, basically recognizes this compound as a fatty acid. You know, if you think about it, H3FTCA is basically a fatty acid where 8 carbon atoms are
Yes, the first three, the carboxylic acid group, and the next two are ordinary hydrogenated carbons, and then the rest are fluorinated. Yes.
Exactly. The organism has these enzyme systems that recognize this as a fatty acid, probably activates it to the CoA thioester, right? And then puts this into glycerophospholipid biosynthesis. And then we find these p-facylated glycerophospholipids in the membrane. And this was an interesting observation, right? When I reported about this, the immediate question was, can you quantify how much is in the membranes? And this is tricky. No standards of p-facylated glycerophospholipid species are available.
No.
And so then we looked into this and realized there are ways of synthesizing these chemicals, but it's not easy to do. And then we decided we ask a company for help, and we now work with a company to synthesize 2 of these p-facylated glycerophospholipid species, but it's difficult. We have one available, the other one is they're still trying to purify it, and it's not cheap, it's expensive. So we have one now, we just got that, and now we are in a position to potentially quantify how much of these fluorotelomere carboxylic acids reside in bacterial membranes.
As they could be a sink. There's far more in the environment than we're expecting because they're hidden in the membranes.
Yeah, so this is one of the implications, right? If you think about analytical workflow to measure PFASs in our groundwater or in soil or in, solids, they would typically miss the fraction of PFASs that are covalently incorporated into biomolecules like membranes. And so there's a potential that the current analytical workflows miss a portion of the PFASs that actually reside in a given environment.
And at the moment, we don't know what proportion that is.
Yeah, so this is something we're working on, trying to quantify and also measure these p-facylated lipid species in environmental samples. Right?
So given that they're in the lipids, can you speculate what they might be doing to the bacterium's physiology?
So that's a good question. So when I give talks about this topic, this is a question that always comes up. What does this do to the membrane of the microbe, of the bacterium? I don't have compelling answers. What I do know, we did compare growing the organism with and without H3FTCAs, and we do not see a difference in terms of growth performance, growth rate, growth yield. You can transfer them many times in the presence of these H3FTCAs. They seem to be doing fine. But it's clear that there's a disruption to the membrane, right? When we did this work with the terminally fluorinated alkanes, when we do the lipidomics, we did see a response of the organism when the acyl chains carried a single fluorine substitution. So for example, the ones that were grown on the fluorinated alkanes, we often saw additional unsaturations. And sometimes we also saw that the other acyl chains that are non-fluorinated were 1 or 2 C2 units longer, suggesting to us that the organism does respond to this fluorination. Now, we haven't done this type of analysis with bacteria that incorporated the H3FTCAs. That's something we still have to do. But it's very clear that the membrane is impacted.
Yeah.
But it appears the organism can respond and is doing fine. I would have to, since I'm not an expert in membranes, but many of my colleagues are very excited and have a research program investigating membranes. So we are starting or have started collaborations with them, and this is a question they're all very interested in answering.
I think that's a fascinating question because doing fine in a culture medium in the lab and doing fine in the real world, of course, might be two very different things.
Oh, absolutely, absolutely. But so far, our working hypothesis is that, so we tested other bacteria, if they can also do this. And so far, we tested maybe 6, 7 different bacteria. They could all do this. Not with the same efficiency, but they could all incorporate these H3 or N3 FTCAs into their glycerophospholipid pool. So it's not a process that's limited to specific bacteria. It appears that probably all bacteria have this capability, or many bacteria.
And what about more wider than that? You said that we have PFASs in our blood. Is it possible that we could be incorporating them into our lipidome as well?
So we have demonstrated this process using bacteria. And so an obvious question then is, can eukaryotes do this too? Right? And so I have a student exploring cell cultures and algae, if they can do this. And this is work that is in progress. I don't have data yet. The bacteria have a different system of incorporating exogenous fatty acids into the glycerophospholipid pool than eukaryotes. That's at least my understanding. But it doesn't preclude the possibility that eukaryotic systems can also incorporate these compounds into their glycerophospholipid pools. So this is a possibility, and it's certainly intriguing to think about the possibility that mammals, maybe even humans, have PFASs covalently incorporated in the lipids of their cells. This is speculation at this point, but certainly somebody needs to look into this because this may have implications. I have no information if this is a bad thing. It could also be a good thing. Who knows?
And you've said as well in your paper that this might be a good thing in terms of bioremediation. This might be a mechanism to mop these things up out of the environment. Can you say how that might work?
Obviously, it's a big goal is to remove these compounds, these PFASs from environmental systems. And so tools have been developed to concentrate PFAS from highly contaminated areas, right? Extract them, concentrate the PFAS, and then have physical chemical methods to destroy the PFAS. This is available, but it only works on relatively small scale. PFASs are everywhere in the environment. And these types of technologies have no impact on the environment at large.
Yeah.
So what we need is a better understanding how microbes interact with these compounds and potentially contribute to removal and destruction. And so now what the process we published in this Nature Microbiology paper does not destroy the PFAS, right? There's no defluorination.
No.
If you think about the organism with fluorinated membranes, it dies, the membrane lies, the ester bonds are hydrolyzed. The N3 FTCAs would be released unchanged. But maybe they're turned over very quickly, right? They're released and go back in new biomass.
Mm.
But then they are actually not really available and are sequestered in biomass. Maybe this is a potential attenuation mechanism, right? Who knows? We have to explore. But people talk about the unique properties of PFASs. I was struck by an image I saw in a publication that describes the fluorescence effect. So they had a nice illustration where they have an aqueous phase, a hydrocarbon phase, and a perfluorocarbon. And this forms 3 separate phases. Okay, so these polyfluorinated chemicals are just different. They don't dissolve in water. They don't dissolve in an organic solvent. They form their separate phase. They like to stick to each other. But there's a possibility that when you have fluorinated membranes, that these bacteria can actually attract or sorb additional PFASs that are in water. So these may be accumulators of PFASs. And maybe one can think about a technology where you use microbial biomass to filter out PFASs from groundwater and then collect the biomass somehow. Right? So there are some ideas that are floating around. This is an example, but I think we are still far away from an application. What we're studying right now is the basic principles. How do microbes interact with these synthetic chemicals, PFASs, and use this information to ultimately come up with a solution that may be workable in systems where these extraction and destruction methods are not applicable.
There's a lot of work still to be done, but this is very much a fascinating topic going forward.
Yeah. So I think this is really nice now to, connect. I mean, I'm interested in bioremediation, but now I have an opportunity, to branch out to people doing lipidomics and studying membranes and connecting this with the ultimate goal, developing an application, a strategy that can be implemented in environmental systems. With the goal, my goal has always been to do fundamental research and make this transition to an application for an environmental system, to clean up the environment, for the betterment of the environment. And I think this offers opportunities.
Well, that's great. We will look forward to hearing a lot more from you in the future. Frank, thanks very much for joining us in the Lipid Maps podcast.
Thank you very much again.
And to those listening, please join us for the next one.
Brought to you by Lipid Maps.